Hello, ladies and gentlemen. Today's mini lecture is on this process called translation. Translation converts an mRNA message from transcription into a polypeptide chain or protein. So we've already been learning a little bit about the central dogma, and this basically states that information goes from DNA to RNA to proteins. We've learned about how replication copies the DNA. We've learned about how to go from DNA to RNA. We have to go through this process called transcription. And today we're going to focus on how we get from RNA to that protein, and that is translation. Now, this can be very hard to visualize, so I've tried to make a helpful analogy for you. First off, let's imagine that DNA is like some sort of a great big recipe book. In this case, the Big Book of Knitting. Tons of different patterns inside, and they all code for something different. When DNA goes through replication, as we've learned, it makes an exact copy of itself. All right? But now, when DNA is converted into RNA, we don't need the whole DNA to be converted into RNA. We just need one pattern, one gene. So it's like copying a single page inside of that book. But now we need to take that information and we need to make something physical with it. In this case, a protein. So that is the process of translation. This is where many different things will actually use that RNA pattern to make a protein. In this case, we're using that pattern to make a pair of socks. Now, as an added bonus, the thing that's doing the making would be a ribosome, and that's kind of like the person who's doing the knitting, so that's kind of cute there. Now, in translation, the language, so to speak, is going to be something called an amino acid. You may remember that in both DNA and in RNA, the language was a nucleotide, but now we need to convert into something else called an amino acid. So translation converts mRNA messages into polypeptides, and all a polypeptide is a chain of these amino acids. Now we're going to introduce you to this idea of a codon. A codon is very important. It's a sequence of 3 nucleotides, and it codes for a specific amino acid. So there are over 20 different amino acids, and basically each different codon will code for a specific amino acid. There are also these things called stop codons. They tell the message to stop turning into a protein. There are 3 of those. And there is one start codon, and that is this guy right here, methionine. So in order to figure out what you're looking for, let's pretend that we've got, UAC. What you're going to do is you're going to start on this side first. You're going to look for the letter U, which I found up here. All right, next I'm going to look across the top for letter A, which brings me right here. And then finally, I'm going to go for the last part. So UAC would be here, and that would be tyrosine. So if I had the codon UAC, it would code for tyrosine. And you guys are going to get more practice with this. So now we're going to talk about reading frames. And basically what that means is a change in the order in which codons are read will change the resulting protein. So here we've got 2 identical strands of mRNA, but you'll notice if we start at the beginning versus if we start one nucleotide off, we get completely different amino acids that it's coding for, okay? And now, regardless of the organism or living thing, codons will always code for the same amino acid. We call this the universal language. So it doesn't matter if you are a bacteria, an earthworm, an elephant, a blue whale, or a human. If you have these nucleotides right here, ACA, they will always code for this amino acid, THR. So amino acids have to get linked together to become a protein. They can't be a protein on their own. And an anticodon is basically a set of 3 nucleotides that is complementary to an mRNA. We find the anticodons on the bottom of the tRNA. So this is an anticodon right here. This is a tRNA or a transfer RNA. And you may remember its job is to carry amino acids to the ribosome from the cytoplasm. So ribosomes are the actual protein building factories, and these guys are really important. They consist of 2 subunits. There's a large upper subunit, which binds to the tRNA, and it has 3 building sites. And then there's a small subunit, and this binds to the mRNA, or the messenger RNA. So what actually happens? Well, for translation to begin, basically tRNA has to bind to a start codon on the mRNA and signals the ribosome to assemble. Remember that start codon is methionine, MET. So a complementary tRNA molecule will bind to the exposed codon, bringing its amino acid close to the first amino acid. So it ends up kind of looking like this, basically. Here's our ribosome. It's clamped onto mRNA. There will be a codon inside of one of these little reading segments, and a tRNA with the correct anticodon goes, ooh, I match. And it comes in and it'll leave the little polypeptide. Another tRNA will read the next frame and it'll come in and it'll bring its amino acid, and they will start to link together with a peptide bond. And if we keep going, you'll kind of notice that one is in the middle, one is leaving, one is coming, and it just keeps on going like that until it reaches one of those stop codons. At that point, the ribosome itself disassembles, and the protein, the polypeptide chain, the amino acid chain, kind of floats out and turns into a protein by kind of bending and flexing. And it looks like that is it for this lecture. If you guys have any questions, please let me know.